So first up is, there we go. First up is Alex Price. So Alex, go ahead.
Okay, hello everyone. I hope you're all keeping well under the current circumstances. So a bit of background on me. I'm trained in biological sciences originally, medical biology, but then retrained in environmental microbiology during my master's over in Scandinavia, where I focused my research project on extremophile microorganisms and microfossils within Antarctic gypsum aggregates. For the last few years since then, I've been doing my PhD in astrobiology with the research group over at the Open University, which is in the United Kingdom. And actually last week, eventually got my thesis submitted, albeit from isolation, which was quite an experience. Okay, so if we could go to the next slide, I'll talk about my project. Cool. So the idea of the PhD was to identify kinds of microorganism that might be able to survive on early Mars in those kind of environments, and also which might leave some kind of distinctive biosignature or record in the Martian geology for us to find today. The way I did that was to use the early Mars environment as a kind of filter. So it's anaerobic, meaning there's not a lot of free oxygen around. So we need to think about other oxidants that microbes can use, things like nitrates and perchlorates that we have on the Martian surface. Additionally, energy sources abundant on Mars are inorganic ones, so things like iron and sulfur, which are really good electron donors for microorganisms. And that brought me to this nitrate-dependent ion oxidation metabolism. So as you can see in the upper central image, this basically involves the oxidation of iron by these microbes, together with the reduction of nitrate to reduce nitrogen species. Now, in the context of early Mars, if you look at the lower central figure, that involves this kind of early CO2 nitrogen atmosphere would be processed by abiotic means to produce the nitrates that we see in ancient sediments on Mars, so we know that were present in the early environment. So we have the nitrates. We would also have dissolved reduced iron being released by dissolution from the bedrock during either volcanic or impact-generated hydrothermal activity. So you have the electron donor and the electron acceptor together in this early environment, along with the CO2 atmosphere, but also endogenous organic carbon that we now know is present on the Martian surface as carbon sources for these organisms. So I've been looking at this experimentally using these really beautifully colored Mars simulant media that are based on the geochemistry of different locations on the Martian surface. Looking at the organism's survival in these, and also the organism's ability to grow on solid iron substrates, so things like olivine minerals. And in this image on the bottom right, this SEM image, you can see one really interesting characteristic of these organisms, which is that under desiccating or high dissolved iron concentrations, they can actually biomineralize. So you get the Fe3 producing oxides, which coats these organisms, encrusts them in iron oxides, laying down a kind of morphological biosignature that may be preserved in the rock record. There's also some evidence to suggest that these can have a protective effect on organics. So you might be able to combine morphological and chemical biosignature detection in these circumstances. Alongside this side of my project, I've been doing a lot of terrestrial microbiology stuff. So doing draft genome sequencing of these organisms, as well as genome analysis. And more recently as well, as part of a follow-on research assistant position in Botswana at a new Mars Hesperian analog site, doing a bit of field work to characterize that as a Mars analog environment as well. So if we could go to the last slide. Cool. So yeah, if any of that sounds of interest to you, either as a collaborator, future employer, whatever, or if you're just interested in the research and want to talk about it, then find me on Twitter, join me in the breakout room, or just drop me an email at some point. Thank you very much for your attention.
Great. Thank you, Alex. We should now be ready for Zoe. Zoe, ready to go?
Yeah, I'm ready.
All right. Very good. Whenever you're ready.
Okay. Hello, my name is Zoe Moreland. I'm a second year PhD student at the Open University, and I'm looking into whether biomarkers could be transferred from Mars to one of its moons, Phobos. My PhD uses the planetary geology knowledge I gained from my bachelor's degree in geology with planetary science at the University of Manchester in the UK, and applies it to one of astrobiology's biggest questions: where might the most likely locations be for finding evidence of life in our solar system? Next slide, please. It's hypothesized that early on in Mars's history, some regions may have been habitable, and that life may have developed and left behind biomarkers. Many past, present, and future missions are going to go and search for evidence of life. However, is Mars itself really the best place to search? If a large impact was to occur on Mars, where biomarkers may be preserved, there is a reasonable chance that some of this ejecta containing biomarkers could be deposited on Phobos, the larger and innermost of Mars's moons. Models have suggested that around 250 parts per million of Phobos's regolith is Martian ejecta. And if this were the case, a mission such as MMX, due to launch in 2024, which aims to collect and return samples from Phobos, could return particles of Martian material as well, which could in turn contain biologically relevant information. To truly understand whether biomarkers could survive the transfer from Mars to Phobos, it's necessary to complete laboratory simulations. Past investigations motivated by planetary protection have looked into extant life surviving the transfer process, because this would raise questions as to whether or not Phobos should be viewed in a similar way to Mars under planetary protection guidelines. However, in my PhD, I've taken a step back to look at the building blocks for life, or the breakdown products of past life, in the form of biomarkers, because these assume less about the forms of life that could have existed on Mars, and they also could exhibit resilience against the harsh conditions of the Martian surface. The aim is to simulate and assess the survival and modification of biomarkers at each stage of the transfer process, from initial impact ejection from Mars through to the ascension through the atmosphere, and then deposition onto the surface of Phobos in a series of experiments using, please next slide, the all-axis two-stage light gas gun at the Open University, capable of firing projectiles a few millimeters in size at several kilometers a second, horizontally, vertically, and anywhere in between. This instrument is perfect to test the survival of biomarkers in hypervelocity impact events. Firstly, I will shoot an inert projectile at more than 5 kilometers a second into a Mars analog doped with biomarkers to simulate a low-speed solar system meteoroid impact onto Mars. Then I will collect this material, the ejecta that comes off, and subject this material to pyrolysis to simulate aerodynamic heating from the ascension through the atmosphere. Then finally, I will make this material into projectiles and fire them into Phobos regolith simulant at a broad range of impact speeds from a few hundred meters per second to several kilometers a second to simulate a variety of possible deposition scenarios on Phobos. Throughout this project, I have become a trusted operator of the light gas gun and will have experimentally crafted unique bespoke projectiles to shoot, whilst also gaining experience in a wide variety of analytical techniques. However, right now, like many, I'm working from home, so I have broadened this project to encompass model analysis as well, to provide an alternative simulation to compare against lab work. The results from this project will provide valuable insight into whether the transfer process from Mars to Phobos is feasible, and if biomarkers do survive the transfer, whether they have been altered in any way. It might then be possible to suggest that Phobos could be better target location for missions searching for evidence of Martian life, rather than Mars itself. Even if this were the case, the concentration of Martian material on Phobos is likely to be extremely low. Therefore, would we really be able to detect them using our current analytical techniques? Furthermore, when we return samples from biologically interesting sites and they undergo sterilization when they return to Earth, will we just be removing the interesting information we're striving to find? If you'd like to know more about what I'm doing and how I'm doing it, please feel free to chat to me in a breakout room after this, And also follow me on social media and take a look at my last LPSC abstracts and look out for papers coming out in the next year. Thank you.
Okay, thank you, Zoe. Okay. All right, Michael. Is Michael ready to go?
Yes, can you hear me?
Yeah, I think I can.
Perfect. Okay. Hi, everyone. First of all, thanks, Andy, and everyone else who put this awesome event together. My name is Mike Wong. I'm a postdoc in the University of Washington's Astronomy Department, Astrobiology program, and the Virtual Planetary Laboratory. My mentor at UW is Vicki Meadows. My scientific interests span the astrobiological gamut from the origin of life to biosignatures to planetary habitability. I was trained as a planetary scientist with a specialty in atmospheric photochemistry. And right now you're looking at some of my thesis work where I modeled the photochemistry of two different Titan states, present-day Titan, which was validated by Cassini data, of course, and an ancient cold methane-poor Titan that we call a snowball Titan by analogy to snowball Earth episodes. You're looking at the downward carbon flux in these pie charts. This is basically the photochemical product that is raining or snowing out onto the surface. And Snowball Titan has about 10 times less stuff total, but also as much, also a much different ratio of hydrocarbons shown in the orange slices and nitriles, which are shown in the lime green slices. And so this helps us explain the missing ethane problem on Titan and also provides a testable hypothesis for future missions like Dragonfly to find evidence of this hypothetical ancient snowball Titan state. So I'm really stoked about Dragonfly and honestly anytime we get to return to the outer solar system. For my PhD at Caltech, I also modeled the photochemistry of Pluto in association with the New Horizons mission. We found out that the beautiful blue haze in Pluto's atmosphere actually plays a really important role in determining the gas phase hydrocarbon abundances in that atmosphere by acting as nucleation sites for the gas to stick to. And I also modeled the atmospheres of early Earth and early Mars with implications for theories for the origin of life. Next slide, please. So currently as a postdoc in the University of Washington's astrobiology program, I'm applying these skills to the frontier of exoplanets. In my current project, we're essentially throwing Venus around different types of stars and asking whether abiotic oxygen can build up from CO2 photolysis. So what you're looking at here are vertical profiles of O2 drawn in solid and ozone drawn in dashed lines for Venus around different types of stars, the Sun, GJ436, and TRAPPIST-1. And what we found out is that the spectral slope of M dwarfs, particularly their high far ultraviolet to near ultraviolet ratio, can drive orders of magnitude more oxygen production. And we're currently in the process of determining if this might be detectable by future telescopes and constitute a false positive biosignature in the search for life. So this is currently work in progress, so keep that in mind with this slide. Next slide, please. Finally, just a word on my most recent published work, which offers a new perspective on the definition of life with implications for astrobiology research, especially in origins of life research. So origin of life theories often take the form of an X first theory, where X might be replication or metabolism or compartmentalization, and they focus very narrowly on the synthesis of super specific building blocks of those mechanisms that we're familiar with in present day life. But my co-author Stuart Bartlett and I acknowledge that this might be a very misleading approach, especially in the grand scheme of things where you start to consider a more universal explanation for the emergence of life throughout the universe. So we created a new vocabulary, life with a Y, which denotes any system that performs 4 criteria of lifeness, namely dissipation, autocatalysis, homeostasis, and learning. And life with an I retains its familiar meaning, denoting life as we know it here on Earth. This classification scheme gives us a new way of understanding life in the context of other phenomena in the universe that share some but not all qualities of life, and new perspectives on seeking life with a Y in the universe, be it de novo in the lab or among the stars. So I've got one more year to go in my postdoc at UW, and will be applying to a wide range of jobs this year. I did apply to be an astronaut last month, but assuming that that doesn't pan out, I will need some form of employment in 2021, be it another postdoc or a permanent position. Besides being at the cutting edge of research, I also really enjoy teaching, mentoring, and public outreach, so a job that allows me to continue those pursuits would be my first choice. If you're hiring or just interested in chatting with me about my work, I'd be happy to meet you. That's all, thanks.
Okay, thank you, Michael. And next up, oops. Oh, Ardith. Did I say that right? I apologize if I did.
That's good, thank you. All right.
Okay, whenever you're ready.
Hello, everyone. My name is Ardith Bravenec, and I am a final year PhD student at the University of Edinburgh, Center for Science at Extreme Conditions in School of Geosciences. Today, I'm going to give you an overview of the two primary components of my PhD project. The first is in planetary science and focuses on volatiles in magma oceans. The presence of water during the early evolution of the Earth and other terrestrial bodies could have had a fundamental influence on their formation, evolution, and potential for habitability. Interior volatile abundances such as hydrogen and carbon may have influenced the development of Earth's oceans, atmosphere, and by inference, the emergence of life. However, much remains unknown regarding the fundamental behavior of water and other volatiles under the extreme conditions of early planetary interiors and magma oceans. The aim of this project is to constrain the hydrogen and carbon budgets of early planetary mantles and moons with their evolving oxidation states, chemical compositions, and thermal histories. For this project, I used novel experimental petrology techniques and thermodynamic modeling. The results of this study detail how water in the early solar system may have behaved very differently compared to present-day Earth. You can go back to the slide, sorry.
Yeah, sorry about that. There we go, sorry.
You can see some of the results in the two figures on the right. In the experimental figure, the ratio of hydrogen and olivine to silicate melt for a very reduced system is much lower than the oxidized system. The second figure is a comparison of models that either account for or ignore redox states and shows a systematic under or overestimation according to redox buffer. These results demonstrate the critical importance of redox in controlling how hydrogen interacts during planetary formation. The second part of my PhD has been in astrobiology and focuses on the preservation potential of molecule biomarkers and organics in planetary subsurfaces. As this figure illustrates, if molecular biotic evidence does exist on Mars, it is likely preserved within or below the crust, where it would be protected from surface radiation. Experimental studies of the interactions between bioorganics and minerals under conditions simulating the Martian environment provide key insights into possible prebiotic processes and the search for life. Okay, next slide. And here is an outline of such an experiment. I used a variety of different starting materials for the study, both natural and synthetic, ancient and fresh biomarkers, such as endolith communities and ancient shells, as well as various clay and sediment samples spiked with biomarkers. I also used bacteria grown in the lab and artificially entombed. This allows us to decipher the influence of different mineral matrices on biosignature preservation. In the second step, these materials are subjected to a variety of different pressure and temperature experiments in both fluid or brine and anhydrous environments, at low or very high temperatures, and up to over 3 gigapascals of pressure. In the third step, after the experiments, the samples are extracted and the extract separated into different fractions based on polarity and derivatized for targeted organic analysis. In the fourth step, instrumental analysis, I used GC pyrolysis for bulk characterization of organics, and LC and liquid GC for trace analysis of biomarkers. ICP was used for the elemental fluid compositions from the brine experiments. And then finally, there's the data analysis. You can see in the figure here at the top that the first chromatogram on the top has numerous peaks indicating different organic compounds. And the last chromatogram, which is the nearly flat baseline, has obviously very, very few peaks. The middle chromatogram, which was subjected to a more intermediate temperature, has retained a variety of organics. And what this suggests is that pressure has produced an initially stabilizing effect and has preserved more organics as a result. All right, thank you. Next slide. Great. Well, I hope you enjoyed it, and I look forward to getting in contact with some of you.
All right. Thank you, Ardith.
Good morning. My name is Carlos Estevez, and I have been working on water on Mars related projects since I was an undergraduate student at the University of Puerto Rico. During my master's work, I have learned how to program in MATLAB as I developed a code to study atmosphere regolith interactions in Mars, specifically by looking at absorption-desorption process. The first step was to combine my skills in physics and astronomy to solve the flux equations for Mars. And therefore, by knowing the difference between the solar energy in and the black body radiation, I could determine the amount of heat that would propagate through the subsurface. In figure 2, you can see how I used Basabada et al. 2017 results and modified the model to account for the rover moving through different types of regolith. In addition to model absorption, I also had to solve the diffusion equations to understand how the water vapor propagated through the surface and the subsurface. Next slide, please. Thank you. With these known parameters, I was able to model absorption for regolith simulants, JSC Mars 1 and Dunite, and see what happens to the near surface humidity cycle. I'm drawn to solve this type of adaptive problems where the solution requires you to learn in the process about a specific subject. So this was the most fun part, as I had to learn about different absorption models and how to code them. As you can see in figure 4, while analyzing the diurnal water vapor cycle along with the diurnal surface coverage, I found that during the night, the regolith could be absorbing humidity from the near surface, while during the day it could be desorbing and acting as a water source. And in my literature review, I found that the same was happening at the polar site in Phoenix in summer. So this might suggest a global phenomenon occurring because they have contradistinct latitudes and different humidity and temperature regimes. In figure 5, you can see the seasonal surface coverage cycle. And we can see that for the spring-summer season, there's a lot of regolith activity alternating between absorption and desorption. And it coincides that during this period of time, in the southern latitudes, the ice sublimates and is able to exchange water with the atmosphere. Meanwhile, in figure 6, we see the subsurface at 10 centimeters of depth, and it seems to be dominated by absorption, as we can see an almost full surface coverage throughout the entire Martian year. And we found out that that absorption could be explaining the observed hydrated 15 centimeter deep layer that was observed by the DAN instrument aboard the MSL. And in figure 7, we can see that our results could imply that the regolith simulants JSC Mars 1 and Dunite may be acting as a water reservoir. And to study this with more depth, I modified the code to keep track of the amount of absorbed water vapor pressure per hour, as you can see in the figure. And our results suggest that the regolith may be absorbing water on a diurnal timescale, such that at the end of the day, you have more absorbed water than you did at the beginning. And in the broad view, the regolith may be absorbing water perennially. And if there were any life under the surface, it could be protected by radiation. So consequently, absorption would be creating a wetter environment and microenvironments in the subsurface that may be habitable to life as we know it. Thus, we suggest future work related on understanding absorption's role to create favorable environments for liquescence processes that could make the Martian subsurface habitable. In addition to solving problems in planetary science, my drive is to apply the acquired knowledge to solve problems here on Earth. Can you move to the next slide, please? Thank you. So during my first semester in the physics master, I developed a technology that was inspired by my previous water in Mars research. The concept behind the idea was to develop a material that was biodegradable and could absorb large amounts of water in a small amount of space, so that we could use this to transport and store water in Mars. But I found that it had many applications here on Earth, including that it could lower the water consumption for farmers, as we could modify the technology to slowly release water. I was able to get experience in the industry, and even got to participate in a competition with a lot of experienced business people. And I won the Student Entrepreneur Award, the Innovation Award, and the third place overall in the competition. And more recently, I decided to use my programming skills this week to help with the actual situation of COVID and help develop a mobile app to tackle the COVID here in Puerto Rico. So if you want to discuss more about my research and my products, you can contact me at my email, carlos.estevez1@upr.edu, and I leave my phone here and my Twitter, quantum_truth. So thank you.
Thank you, Carlos, and thank you to all our presenters this morning. That wraps up our morning session on astrobiology and exobiology.